Okay, so first we have basic principles with regards to immunology. First of course, we have to understand the nature of our immune system. So our immune system is divided into two. We have the innate immunity and we have the adaptive immunity. So the innate immunity, this one is uh, naturally present at birth. That's why tinatawag din siya no, na natural immunity. Okay? And this type of immunity does not have immunologic memory. That's why the other name also for innate immunity is that it is non-specific, no? Because it does not possess immunologic memory. So that means any foreign object or any foreign invaders na pumapasok sa katawan are being attacked, no, by our natural immune system. Now, the natural immune system or the innate immunity covers the first two line of our defenses, no, the first and the second line of defense. So when we say the first line of defense, that refers to our physical barriers, not just that, pati yung ating mga secretions, okay, or even our normal flora that are present in our intestines, these are all part, no, of the first line of defense. Okay? And usually, no, whenever the first line of defense are because this is the lowest form, this is the lowest form of the line of defense. So whenever it is I mean the foreign invader passed through, no, the first line of defense, then that's the time, no, that the second line of defense will take into action. So the second line of defense, still part of the natural immunity. Okay, this refers to those substances that are naturally present in our blood, no? And it is divided into cellular and humoral component. Okay? So for the cellular component, that would be of course, no, yung ating mga phagocytes. Okay? So that is the cellular component of the second line of defense. Okay? So kung tatanungin tayo yung ano daw yung major component, no, of the cellular. Okay, so that the cellular component of the innate immunity, so that would be our phagocytes. So you have there a list, no, of the different phagocytes, and of course alam naman natin, no, kung paano yung kanilang function. Mainly they are able to phagocytize, okay, foreign invaders, no, sa ating katawan. Then kung merong mga cellular components, we also have our humoral components. Okay, so the humoral components naman, ito yung mga soluble factors. Okay? So the main component or the main humoral component of the second line of defense is the complement cascade. But take note here, no, na naka-emphasize dito that it is only the alternative cascade. Because always remember that the classical cascade requires antigen-antibody interaction before it is activated. Okay? And because of that, the classical cascade is part of the adaptive immunity. Okay? So that is, no, yung ating innate immunity, no. So meron tayong first line of defense, basically yung ating physical barriers, the gastric secretions, the sneezing and coughing reflexes, the normal flora in our intestines, even the sweat, no, yung pH ng ating skin, no. All of these are part of the first line of defense. For the second line of defense, yan naman yung divided din, no, into humoral and cellular component. And for the humoral component, the main component there would be the complement. And for the cellular component, that would be the phagocytes. Okay? I'll just point out, no, another humoral component, no, for the second line of defense. Yan yung tinatawag natin na interferon. Okay? So yung interferon, this is a part also of the humoral component of the second line of defense that works, no, on preventing viral replication. That's why most of the viral infections, no, na nakukuha natin, these are self-limiting, no, because of we have this natural component, no, of our immunity, no, that prevents them from replicating. Okay? Now, The highest form of defense would be our third line of defense. And pag sinabi nating third line of defense, yan na yung ating adaptive immunity. Okay? So third line of defense. Now, yung adaptive immunity natin, obviously, this one is underdeveloped at birth. Okay? So kaya siya tinawag na acquired, no? Acquired immunity because it is underdeveloped at birth. And ito yung type of immunity with immunologic memory. Therefore, tinawag din siya na specific, okay? Specific immunity because it already has a immunologic memory. Okay? So, same no with the second line of defense because the third line of defense is also found in our blood. It is also divided into cellular and humoral components. Okay? And when we say cellular components, of course, this refers to our specialized lymphocytes. That would be our T cells and our B cells. Okay? And yung humoral component naman, okay, that would be the product no, of these cells. So, for example, yung ating mga B cells, no, whenever they are activated, they become plasma cells, remember, and sila yung nagpo-produce ng mga antibodies. Okay? And T cells, no, also whenever they are activated, they are producing certain cytokines. Okay? So again, pag sinabi nating cellular component, we are referring to our specialized lymphocytes and yung kanilang mga products that are dissolved no, in the blood, these are humoral components. However, no, if we are really to look into the mechanism of how these lymphocytes are protecting us no, from foreign invaders, basically yung ating T cells, they are the one that is working sa cellular immunity. Kasi majority ng T cells natin, yung kanilang way no, of combating infection is most of the time through direct cell-to-cell interaction. Okay? That's why kung tatanungin tayo, no, sino yung mediator talaga no, ng T-cell immunity? Okay? Sino talaga yung kumbaga bida no sa kumbaga bida sa cellular immunity? That would be our T cells. Kasi yung mga B cells natin, they are only able to elicit their function no in our immune system through the production of antibodies. And antibodies, this is yung antibodies natin are the major component of our humoral immunity when it comes to the acquired immune system. Okay? So yung antibodies, yung major component no of the humoral adaptive immunity. Okay? So actually, we have several ways no in order for us to be able to acquire immunity no. I-segue ko na lang no, hindi ko siya nilagay diyan sa notes. Okay? Segue ko na lang no yung ways natin no para makakuha tayo ng tinatawag na acquired immunity. Okay? So we could get that through natural means and of course through artificial means. And through this means, pwede na tayo mismo yung nagpo-produce ng antibody or we are only given the antibodies. Okay? So having said that, meron tayong 4 na uri no, ng acquired immunity. Okay? So we have yung natural, of course, na way of getting it. And of course, we have the artificial. Okay, so pag natural, pwede guys na natural active. So pag sinabi natin na natural active, we are the ones na nag-produce ng antibody. So that means we are exposed to antigens naturally, kaya tayo nakapag-produce ng antibody. Okay? So example of a natural way of acquiring immunity is through infection. Diba? Kasi once na-infect tayo, diba? Nakakapag-produce tayo ng antibody against those antigens responsible for the infection. Okay? So tayo mismo yung nagpo-produce ng antibody. Pag natural passive naman, okay, that means no, we are only given antibodies through a natural way. So remember, no, we have IgG, no, that could cross the placenta, no, during gestation. So that is an example of a natural passive, no, transplacental, no, IgG transfer, okay, from the mother to the fetus, no. So that is a natural way of acquiring passive immunity, no. So parati lang tatandaan dito na passive, pinasahan ka lang, no, ng antibody. Okay, now we also have natural means, no, of acquiring immunity. Pag natural active, kumbaga merong pag artificial active rather, kumbaga merong consent. Okay, you are given an antigen in order for you to produce an antibody. So this is oftentimes, no, ito yung principle, no, behind, no, the vaccination na program, 'di ba, or the vaccination approach wherein we are given an antigen in order for us to elicit an immune response against it. Okay? So that is an active artificial kasi hindi naman siya natural talaga na nakuha mo yung antigen, no? So kumbaga merong consent. Okay? Then we have artificial passive. Okay? Ito naman yung mga immunoglobulin na mismo yung mga binibigay sa iyo. So for example, we have the hepatitis B immunoglobulin, okay? Rogam for example, 'di ba? Kasi that's already an antibody that is only given to you. Okay? Now, of course, no, kung titingnan natin holistically, mas okay talaga, no, na nakukuha natin yung immunity in an active manner. Because the antibodies yung mga antibodies that are produced, no, by ourselves mismo, these are the type of antibodies that will stay longer, no, in our body. Minsan nga merong mga infection that will provide us with lifetime immunity. Example, yung hepatitis A. If you had hepatitis A, that will give you lifelong immunity. You cannot have hepatitis A anymore. Okay? So mas okay talaga na ikaw yung nagpo-produce ng sarili mong antibody. However, no, if there is one advantage, no, na pwede nating ma-attribute dito sa ating passive means of acquiring immunity, that is readily available kasi yung antibody. I think we could all relate to this, no, nitong early phases, no, of COVID wherein yung vaccine is not yet available. 'Di ba naghahanap ng convalescent plasma ba yun or convalescent serum? Okay? So the point, that is an example of artificial passive because you are providing the patient, no, with a serum coming from a person that was infected. Okay? So binibigyan mo siya mismo ng antibodies, no? So that is an artificial passive type of immunity. Okay? So yung advantage nito, readily available yung antibody. So it could elicit its function right away. Okay? Pero mas okay pa rin, holistically, mas okay pa rin talaga if you are the ones who produce your own antibody. Okay? So yan yung mga means, no, of acquiring immunity. Okay? Now, one of the important, okay, components also of our natural immunity are our acute phase reactants. Okay, so acute phase reactants, this is a humoral component, no, humoral component ito, okay, of natural immunity. Okay? So yung ating mga acute phase reactants, most of them are produced in the liver, okay, driven by interleukin 6. Okay? So yung interleukin 6, ito yung nagda-drive, no? Okay, drives acute phase reactant production. Okay? So, in general, no, yung definition natin for our acute phase reactants, these are substances, no, that are increasing in concentration whenever there is infection. Okay? So, pwede na yung function nila is to protect us against the offending agent, but sometimes meron din silang protective function. Because remember guys that whenever we have inflammation, inflammation is a non-specific response of our immune system. Okay? And we cannot deny that sometimes there are collateral damages, no, caused by inflammation. So minsan, no, yung function din ng acute phase reactant is to be able to sweep off, no, the excessive effects of inflammation. Okay? So here I have enlisted, no, yung mga common, no, na acute phase reactants. The first one, of course, yung pinakakilala, we have C-reactive protein. Okay? So C-reactive protein is an technically, no, formerly actually yung C-reactive protein was mistaken to be an antibody because it was first found, no, attached or reacting to the C substance, no, on the surface of a pneumococci, and kaya siya tinawag na C-reactive protein. Okay? But later on, no, because of course structural differences, no, it was found out not to be an antibody. So si C-reactive protein, this is the most sensitive, no, among all our acute phase reactants. It is the most sensitive because it is able to increase rapidly with levels of up to 1,000 times higher, peaking at 48 hours upon after the onset of inflammation. Okay? So sensitive siya, no? It is the most sensitive acute phase reactant. However, it is non-specific. Whenever your C-reactive protein is increased, you cannot point that to a single disease lang. Diba? So sensitive but non-specific siya na marker of inflammation. Okay? Paki-take note sensitive, okay? Pero non-specific indicator siya of inflammation. Next, activates monocytes and macrophages producing inflammatory molecules. So this is another acute phase reactant that is also increasing 1,000 times. Pero medyo nahuhuli kasi siya as compared to C-reactive protein. Okay, so usually yung peak niya umaabot ng 24 hours whereas nasa yung C-reactive protein kasi, nagsa-start na siya to increase within 4 to 6 hours and nagpipeak siya within 48 hours. Yung amyloid kasi, yung serum amyloid A, no, is nag-e-increase siya, no, within 24 hours after inflammation, no. So although it is also increasing 1,000 times, pero hindi siya ganoon ka-sensitive as compared to C-reactive protein kasi medyo huli siya sa pagtaas sa kanyang level, no. So this is serum amyloid A. Okay? And I would like you to take note also, no, that it has a high affinity with HDL, no. Sa katunayan, high density lipoprotein is considered to be the transport protein of serum amyloid. Next, functions in opsonization, chemotaxis, and lysis of cells. No, yung pinaka-importante dito, no, yung lysis of cells. Alam natin that this is, no, the end product of complement activation, yung lysis of cells. No, so we have our complement. Okay, so we have a separate topic for that later, no, yung ating complement cascade. Next, we have limits, no, the harmful effects of the immune system by inhibiting proteases released from leukocytes and regulating expression of pro-inflammatory cytokines. This is the one that I am emphasizing earlier, that sometimes these acute phase reactants are sweeping off, no, the harmful effects of inflammation. No? So this is alpha-1, okay? Alpha-1 antitrypsin. Okay? So pakilagyan na yan ng star because this is another favorite question in the ASCP exam, no, na yung parating recall question dito is ano daw yung sakit, no, na pwede kang magkaroon if you have a deficiency of this substance, no. So take note, no, that it protects us from the proteases that are released by leukocytes during inflammation. Yung nangyayari kasi, no, whenever there is infection, for example, syempre yung ating white blood cells, aligagayan sila, no. They will do everything in order to eliminate, no, the offending agent, no. So nagre-release sila ng substance, tawag natin doon elastase. And this elastase, this is an example of a protease that is released by white blood cells. Yung elastase, yes, it could kill the offending agent, but it is also non-specific. So whenever elastase is excessive, no, in our circulation, it could destroy normal cells in the body. And that is the work of alpha-1 antitrypsin. okay, to sweep off the excess elastase. Okay? Now, what will happen to you if you do not have alpha-1 antitrypsin? Elastase will destroy normal cells in your body, specifically the cells in your lungs, the cells in your liver. That's why alpha-1 antitrypsin deficiency could result to AAT deficiency. Paki-side notes na lang dyan. Okay? This could result to pulmonary emphysema. Okay? Pulmonary emphysema. And pwede din yung juvenile onset na cirrhosis. Okay? Juvenile cirrhosis. That is due to the destruction of the normal cells, no, in the liver and the lungs, no, because of the excessive effects of inflammation. Okay? Next, we have an antioxidant that binds irreversibly to free hemoglobin, that is haptoglobin. Okay? Yung point lang naman dito, sometimes there are infection, no, that results to lysis, no, of our red blood cells, di ba? And yung hemoglobin, that is toxic to our kidneys. And haptoglobin binding it is actually a mechanism of protecting our kidneys from the toxic effects of hemoglobin. Okay? Siyempre, no, part of it also is to conserve the iron that is present in hemoglobin. Okay? Then, promotes clot formation, that is fibrinogen, no? Which is the most abundant of our coagulation factors. And yung pinaka-purpose of why fibrinogen is also an acute phase reactant, kasi yung clot, it could function by trapping, no, itong mga foreign particles or itong mga offending agent. Pwede na siya yung mag-trap nito, okay? As a result, no, na prevent yung further dissemination of the infection. Okay, and lastly, we have the principal copper protein, copper transporting protein in the plasma, that would be ceruloplasmin. Okay, so si ceruloplasmin also has a function, no, by converting iron, no, from its ferrous to its ferric state, no. Kasi the ferrous state of iron is also toxic. So kino-convert niya ito sa kanyang ferric state, no, para also to prevent, no, the toxic effect, no, of that form of iron. Okay? So those are, no, the acute phase reactants. Again, majority of them are proteins. Actually proteins yan silang lahat. They are increasing, no, whenever there is inflammation. Pero take note that not all proteins are acute phase reactants. Okay? We have proteins that are decreasing their level whenever there are inflammation, no? So sino yung mga proteins na ito? Tawag natin sa kanila are negative, no? Negative acute phase reactants. Okay, negative acute phase reactants, what are they? Okay, number one, of course, no, yung ating albumin. Okay? Then we have prealbumin. Okay, prealbumin, or otherwise known as transthyretin. Okay, and we also have transferrin. Okay, so these are the proteins that are decreasing their level whenever merong inflammation. No? So mga negative acute phase reactants yung tawag natin sa kanila. Okay? So that's it for our acute phase reactants. Another important component of our natural immunity would be our phagocytes, 'di ba? Sila yung major component naman, no? Sila naman yung major cellular component of the natural immunity. Yung mga phagocytes. Okay, of natural or innate immunity. Okay? So take note lang natin no, yung general steps din. The general steps no for phagocytosis. First, of course, we have initiation. And initiation is made possible through yung mga toll-like receptors, TLRs or toll-like receptors that are usually present no sa uh, surface no ng ating mga phagocytes. Okay? And these receptors, ito yung nakakapag-recognize ng mga uh, uh, pathogen-associated molecular pattern. Sorry. So yung mga PAMP, no? Pathogen, okay, pathogen-associated molecular pattern. Okay, so nare-recognize ito no ng ating mga phagocytes, and because of that no kumbaga ina-attack nila no ina-attack nila yung mga pathogens. Okay? So again, no, through the TLRs, no, yung mga PAM or pathogen-associated molecular pattern are being recognized. Okay, so nai-initiate yung phagocytosis. Next step is diapedesis and chemotaxis. No, so when you say diapedesis, this is the migration of phagocytes from the blood vessels, capillaries to the injured tissue. So kumbaga, remember our phagocytes are present inside our blood vessels, right? So they are to cross no the vessel wall towards the site of infection, and that process is diapedesis. Okay? Or the extravasation, no extravasation of phagocytes through the vessel wall. Then they will now start to move towards no the site of infection. This is made possible also by certain substances, tawag natin doon chemoattractants. Itong mga chemoattractants, they are attracting, no? Obviously, they are attracting phagocytes no, towards the site of infection. So itong process na ito is chemotaxis. Okay? So syempre, no, when the phagocyte is already present there in the site of infection, it will start to engulf no, the offending agent, and it has to elicit no, a very important process. Yung tawag natin doon is respiratory burst or oxidative burst. No? Respiratory burst or oxidative burst, because this mechanism will kill the offending agent. Okay? It's not enough that the phagocytes are able to engulf the offending agent, but they also have to digest it. And minsan, nagkakaroon ng problema in the process of digestion, because there are phagocytes that cannot, there are neutrophils to be specific, that cannot digest, or they have the inability to generate an oxidative burst. And because of that, it would result to a certain disease. Ano tawag natin sa sakit na yon? Okay? Ano tawag natin sa sakit wherein our neutrophils cannot mount no an oxidative burst. That's why if you have this, nagkakaroon ng recurrent no severe, okay, severe recurrent bacterial infection. Okay, because our phagocytes, yes, they could engulf the offending agent, but they cannot digest it. Okay, so tawag natin sa group of disorders na ito is chronic. Okay, chronic granulomatous disease. Okay, chronic granulomatous disease. So again, this is a group of disorders characterized by a defective NADPH oxidase system. Okay? And itong NADPH, this is a very important enzyme no in the process of phagocytosis. Kasi there are two ways no in order for the phagocyte no to digest or to kill the offending agent. Meron kasi tayong tinatawag na oxygen-dependent pathway and oxygen-independent pathway. And in both of those pathways involve yung NADPH oxidase. For example, In the oxygen-dependent pathway, your NADPH oxidase, take note lang natin yung function niya. Okay? In the oxygen-dependent pathway, your NADPH is capable of sulat ko muna si NADPH. Okay, NADPH oxidase. Ano ba yung function nito? Bakit sobrang importante nito? Because si NADPH oxidase, it is capable of reducing, no? So it reduces oxygen, okay, to superoxide. Okay, so this process is in the oxygen-dependent pathway. Then later on, si superoxide, with the aid of superoxide dismutase, it will be converted into active oxygen radicals. And these oxygen radicals will kill the pathogen. Okay, so that gives us the idea na hindi din na-mount yung respiratory burst or hindi tayo nakakapag-produce ng reactive oxygen species if NADPH oxidase is absent. Okay, now, again, NADPH oxidase is also involved in the oxygen-independent pathway. How? Because NADPH oxidase depolarizes, okay, it depolarizes the membrane, okay, of our phagocytes. Okay, it depolarizes the membrane of our phagocytes. And because of this, no, this process kasi of depolarization is very important in order for potassium ions to enter the cell. And once potassium ions are entering the cell, no, it will basically alter the pH inside, no, the body, no, or inside the cell. And this change in the pH will result to activation of certain enzymes. Tapos itong mga enzymes na to will digest the pathogen. So yun naman yung oxygen-independent pathway. Yung target mo dito is to activate digestive enzymes. And in order for you to do that, you have to depolarize the membrane, which requires also NADPH oxidase. That's why si NADPH very important. Kailangan siya in the two pathways of killing, no, the offending agent. Now, chronic granulomatous disease, you have a defective NADPH oxidase system. Therefore, nagkakaroon ka ng mga, you are characterized, no, by severe recurrent infections. Yan. Okay? Now, I think also important is to take note of the tests for chronic granulomatous disease. Okay? So we have the classic one, yung tinatawag natin na nitro-blue. Okay, nitro-blue tetrazolium test. Okay? So itong si nitro-blue tetrazolium, this is naturally a colorless or para siyang yellowish to colorless na dye, si NBT. No? This is a originally para siyang yellow, no, yellow or colorless na dye. Okay? Now, when NBT is reduced because of the production of H2O2, it forms a blue formazan dye. Okay? Now, hindi ka naman makakapag-produce ng H2O2, di ba? If you do not have NADPH oxidase. Okay? So, pag hindi ka so that means, no, the positive result here is the presence, no? The presence of the blue color. Okay? Kung meron kang blue color, ibig sabihin okay yung mga neutrophil mo. They are able to mount a respiratory burst. Okay? So that's the nitro-blue tetrazolium test. However, no, we also have the flow cytometric assay for this. Okay, flow cytometric assay. Okay? So dito naman, no, we are using a label for our neutrophils, no? So yung label na ginagamit natin, take note lang, is dihydrorhodamine. Okay, dihydrorhodamine. And of course, we have to activate the neutrophils. So how do we do that? Yung ating activator, okay? Our activator is 4-ball. 4-ball myristate acetate. Okay, 4-ball myristate acetate. So that is the activator. So when your neutrophils are activated, of course, they will fluoresce. No? Nagkakaroon ng fluorescence if na-activate, no, yung mga neutrophils. And that is the positive result. Ibig sabihin, nagkakaroon ka ng oxidative burst if there is fluorescence. And yung fluorescence, it is being quantified through flow cytometry. Okay? So yan naman yung flow cytometric assay, no, as a test, no, for CGD. Again, emphasize ko lang yung positive, yung result dito, if merong, no, if oxidative burst is present, no, if oxidative burst, okay, is present, okay, magkakaroon dito ng blue color production, di ba? For the nitro-blue tetrazolium test. Dito naman, if oxidative burst is present, okay, in the flow cytometric assay, no, magkakaroon ng fluorescence. And the fluorescence is quantified with the use of the flow, flow cytometry. Okay? So let us now proceed to the different lymphocytes. Okay? So yung lymphocytes, sila naman yung mga bida kumbaga, no, sa ating adaptive immunity. Okay? So we have three populations for lymphocytes. We have T cells, okay, comprising approximately 60, no, to 80%, no, of the lymphocytes in our blood. Okay? We have B cells na nasa 10 to 20%. Okay? And we also have natural killer cells na nasa 10 to 15%. Okay? So first, no, we have our T cells. They are involved, no, sa tinatawag natin na cell-mediated immunity, no? So they are able to elicit their function through cell-to-cell interaction, no? Direct cell-to-cell interaction. Okay? Yung product nila would be our cytotoxins, no? Including perforins and granzymes, and they are good in defending us against viral fungal infection, also involved in tumor rejection and in graft rejection. Okay, yan yung mga T cells. Very important dito no yung mga surface markers. Okay, ay ano talaga yan? Rampant din yan no sa ASCP exam no yung mga surface markers, because these are our ways no in order for us to identify a particular cell population. And medyo mahirap no yung mga CD markers. Okay lang kung sa lymphocytes, pero medyo mahirap siya when it comes to leukemia, no? Sa leukemia kasi napakarami. So anyway, no, pag-usapan natin 'yan pag narating na natin yung hima. Okay, so first, surface markers muna tayo no for our T cells. Okay? So T cell surface markers. Okay? So number one, we have our CD2. Yung CD2, ito yung tinatawag na rosette. Okay? Receptor. Okay? Papag-usapan natin yan mamaya, yung rosette test. Then we have CD3. CD3 is a component. Hindi siya yung mismong T-cell receptor, but it is just a component of the T-cell receptor. Or yung TCR. Okay? Then we have CD4. Of course, yung CD4, ito yung nakikita natin sa ating T helper cells. Okay? So mature na to, no? This is for the mature T marker na to. Then we also have our CD5. Then we have CD7. Yung CD5 tsaka CD7, these are present, no, in immature T cells. Okay? And lastly, we have CD8, no, which is found in our T cytotoxic cells. Okay? So, syempre, mature marker na din siya. Yan. So, these are the markers, no, for our T cells. Okay? And if you would notice, no, isa sa mga ginagamit na palatandaan dito, medyo maliliit na mga numbers sila, no? 2, 3, 4, 5, 7, 8. 'Di ba? Maliliit na numbers, no? So, that is for T cells. Okay? Now, we already mentioned, no, earlier that yung ating B cells, no, is yung involved talaga sa humoral immunity because they are able to produce antibodies, no? You have there in the notes. So, yung mga B cells naman natin, they are good in defending us against bacterial infection through, again, no, yung mga product nila which are our antibodies. And of course, no, pag-uusapan din natin kung sino-sino yung mga B cell, okay, surface markers. Okay, B cell surface markers. No? So first, of course, we have the IgM and IgD. Okay? Meron silang immunoglobulin M tsaka immunoglobulin D. Pag hindi pa sila naging plasma cell, of course. Kasi yung IgM tsaka IgD, guys, ito yung counterpart ng T cell receptor. Kumbaga, yung ating mga B cell, they are able to attach to an antigen through the surface antibodies. No? So kumbaga, ito yung nagsiserve na receptor nila. Okay? Then we have CD21. No? Yung CD21, this is the Epstein-Barr virus receptor. Okay? So this gives us the idea na yung ating, yung Epstein-Barr virus, meron siyang kumbaga yung cell na ini-infect niya are the B cells, no? Kasi yung receptor niya is the CD21 which is found in B cells. Okay? Then we have CD10. CD10, ito yung tinatawag natin na KALA, no? Yung common, okay, the common acute lymphoblastic leukemia, yung ALL, no? Common acute lymphoblastic leukemia antigen. Okay? Then we also have CD19, 20, and 22, of course. Okay, CD19, okay, sulat ko na lang dito CD19, CD20, CD22. These are the markers that are often found in mature B cells. Okay, and syempre meron din siyang MHC class 2. Okay, meron siyang MHC class 2. So these are the markers, okay, that can be found, no, sa ating B cells, no. So if you would notice, medyo malalaking numbers sila. Kaya nga sabi nila B daw big, no? Big numbers. Okay? T daw tiny kasi maliit, small numbers. Okay? Then we go to our natural killer cells. Okay, so paki-fill in na lang dito yung mga nasulat natin na markers. Okay, so we have our natural killer cells, nasa 10 to 15% sila, okay, ng ating 10 to 15% sila ng ating lymphocyte population. Involved naman sila sa antibody-dependent cellular cytotoxicity. Yung natural killer cell are the only lymphocytes that are part of our natural immunity. Again, they are the only lymphocytes that are part of our natural immunity. So, they are also able to produce cytotoxins, katulad ng mga T cells, and they are good in defending us against virus-infected cells and tumor cells. And there are two surface markers, no, for natural killer cells. First, we have the CD16, okay? the CD16 which actually serves as its receptor, no? Ito yung kanyang IgG receptor. Okay? And we also have CD56. Okay? Pakilagyan ng star, recall question yan during my exam. The question was, meron daw cell, no? Meron daw cell and the cell was found to be negative for CD2, CD3, CD19, CD20, parang ganun. Negative daw yung cell, no? Negative daw siya para sa CD2, CD3, CD19, tsaka CD20. Anong cell daw siya? The choices were T helper, T cytotoxic, B, tsaka natural killer. So syempre, no, the answer is natural killer cell. Kasi wala naman talaga siyang CD2, wala siyang CD3, wala siyang CD19, wala siyang CD20. Okay? So, yan, no? May mga basic din sila, mga ano din, mga easy din na questions, no, paminsan-minsan. Okay? Pero pag in-apply na yan nila sa flow cytometry, ay, ayun na. Yun na yung medyo mahirap. Okay, so mitogens, no, these are substances that are used, no, in order to kumbaga mitogens, no, in order to paramihin mo yung lymphocytes. Okay? So we have phytohemagglutinin, concanavalin A, and poquid mitogen, no? Good, ginagamit siya, no, for T cells. Then we have the poquid mitogen and staphylococcal protein A naman, no, for the B cells. Okay? Now, pag-usapan natin, no, yung maturation stages of lymphocytes. So first, we have the B cell maturation muna. So the first step here is characterized by rearrangement of genes coding for heavy chains and light chains. First, take note muna natin, saan ba nakikita yung genes na nagko-code for heavy chain? That is found in our chromosome number 14. Okay, chromosome 14, yung heavy. Okay, tapos yung light chains naman nakikita natin sa ating chromosome 2 and 22. Dito naman nakikita yung mga domains or yung mga loci rather, that codes for the production of light chains. So take note, in the first stage, which is the pro-B cell, no, the pro-B cell stage, take note, hindi ka pa nagpo-produce ng heavy chains and light chains. You are just rearranging, no, the genes that will code for them later. Okay, so take note, no, what are the distinctive markers of pro-B cell? We have CD19, okay, we have CD45R, we have CD43, CD24, and the SIKIT. So these are the distinctive markers of pro-B cell. No, yung term na pro-B cell, guys, that means progenitor. No, progenitor B cell. Okay? Next step is yung ating pre-B cell, of course, no? The pre-B cell. Pre naman means precursor. Okay, precursor B cell. And this is characterized by appearance of mu chains. So yung mu chain, this is a heavy chain, no? So nagkakaroon na ng appearance ng mu chain. And it is but proper na yung pinakaunang immunoglobulin, no? Yung pinakaunang immunoglobulin that is found on the surface of a developing B cell is IgM. Kasi heavy chain niya yung una mong napoproduce. Okay? Heavy chain niya yung una mong napoproduce, no? So pag nagkaroon na ng IgM, no, sa surface ng developing B cell, tawag natin sa kanya is immature. So that is now an immature B cell. So take note, it is characterized by presence of a surface immunoglobulin M. And this immunoglobulin M is a monomer. Okay, di ba naturally yung mga IgM natin pentamer pag sa dugo. Pero pag nasa surface sila ng B cell, monomer yan sila. Okay? Now, what are the distinctive markers of immature B cell? So meron na siyang CD21, CD40, and MHC class 2. Okay? So, alam naman natin na yung MHC class 2, that is necessary for antigen recognition. Kasi guys, take note natin that it is in the immature B cell stage kung saan nagsa-start yung tinatawag natin na selective process. Okay? So I will elaborate that later, no? Yung tinatawag na positive tsaka negative selective process. Positive and negative selection process rather. Okay, positive and negative selection process. Okay? Then, after the appearance of IgM, susundan siya ng appearance of IgD. So IgD will now have IgM. Yung next stage, which is the mature B cell, no? Mature B cell will now be characterized by the presence of IgM and IgD at the same time. And syempre, no, meron din siyang MHC class 2. No? Pakidagdag na lang, no, yung MHC class 2 kasi kailangan again yan for antigen recognition. Okay? Then, the next stage is an activated B cell. Okay? So, nakikilala natin guys yung isang B cell na activated na siya pag meron na siyang CD25. Because the CD25, it will serve as the receptor for interleukin 2. Okay? And when interleukin 2 will now influence a B cell, it will either become a memory B cell or a plasma cell. And yung plasma cell, we all know, sila yung nagpo-produce ng antibodies. So take note guys na yung ating mga plasma cells, they no longer have surface immunoglobulins. Again, they do not have surface immunoglobulins anymore. Instead, yung kanilang mga immunoglobulins are found in the cytoplasm. No? So cytoplasmic immunoglobulins. Okay? So the other term for the plasma cell is effector B cell. Okay? The other term naman for immature B cell is competent, no? Competent B cell, no? Kasi may ano din may recall question din to dati. Yung tanong daw is which of the following is true with regards to competent B cell? Parang immunocompetent B cell. So if they are immunocompetent, that means they are already able to bind an antigen. No, nakakapag-bind na sila ng antigen. And they can only do that if they have the surface immunoglobulin. That's why nga meron tayong tinatawag na selection process kasi part ng selection process is to check kaya niya na bang mag-bind ng antigen. Okay? If not, pinapatay. Okay, namamatay itong B cell. Okay? Next, we go to the steps naman for our T cells. Development naman ng T cells. Okay, so first, ito medyo easy lang yung mga T cell development stage. Titingnan mo lang if whether they are CD4, CD8 negative, positive, or positive lang siya sa isa. I mean negative for both, positive for both, or negative lang siya or positive lang siya either sa CD4 or sa CD8. So the first stage is yung tinatawag natin na double negative thymocyte. Okay, double negative thymocyte. The reason for that is that it is both negative for CD4 and CD8. However, it already has CD2, CD5, and CD7. Okay? Question, sige nga. Can this double negative thymocyte bind an antigen already? Kaya niya na bang mag-bind ng antigen? Hindi pa, no? Hindi pa. Dahil it is in the double positive thymocyte stage where the selection process starts. Why? Because remember na yung CD3 is a component of the T cell receptor. And yung double negative thymocyte, wala pa siyang CD3. So technically, it cannot bind an antigen yet. Okay? So the selection process starts in the double positive thymocyte stage. Yeah. Okay? So meron na siyang CD3, CD4, and CD8. Thus, this is where the selection process is happening. Okay? Then we now have mature T cell, and yung mature T cell natin can only be CD4 positive or CD8 positive. Okay? So nagiging CD4 positive siya if it recognizes an antigen along with class 2 MHC. Nagiging CD8 siya if it recognizes antigen along with class 1 MHC. Okay? And again, pag meron ng CD25, yan yung tinatawag natin na activated lymphocyte, no? So activated T cell, no, in this case. Kasi again, yung CD25, it serves as a receptor for interleukin 2. Okay? So yan po, no, yung mga stages, no, for T cell maturation. Now, let us proceed, no, sa laboratory identification of lymphocytes. So, this is through flow cytometry, no, pero as I mentioned earlier, no, idadagdag ko dito yung Rosette test, no, or yung Rosette technique kasi classic kasi siya na test for identification din, no, of lymphocytes. So yung tinatawag natin na Rosette test. So just a few consideration. Okay? So here, gumagamit tayo ng sheep red blood cells, di ba? And yung sheep red blood cells natin, okay, this will attach, no, to the CD2. Okay, CD2 antigen that is found in our lymphocytes. Okay, so considering that it is only attaching to the CD2 antigen, this gives us the idea that your Rosette test cannot be used to enumerate, no, or at least enumerate B cells. Kasi walang CD2 yung B cells, no? So this can only be used, no, for T cells. Okay? Then in order for you to consider it as a Rosette, okay, dapat at least, no, para maging matawag mo siya na Rosette, dapat at least, okay, at least 3 red blood cells yung nakadikit sa lymphocyte, no, or sa T cell na yan. Okay? So yung gagawin mo dito, you are to count 200 cells, okay? Are counted. Okay? And you solve for percent forming rosettes. Okay? Percent forming rosettes. So example, no? Let's say, sa 200 na T cell na na-count mo, okay, 50 sa kanila, for example, yung nagro-rosette. So that is simply 50 over 200 times 100. Diba? That is now your percent. That is now the result for your Rosette test. Okay? So 200 cells yung ika-count mo regardless if whether may nakadikit sa kanya na RBC o wala. No? So only those na may nakadikit na at least 3 RBC yung ika-count mo as a Rosette. Okay? So that is the Rosette test. Next. However, no, yung pwede nating gamitin, no, that could be useful in measuring both T and B cell would be flow cytometry. So this technique is rapid and convenient for generating immunophenotypic data, and this is useful, no, for the evaluation and prognosis of leukemia, lymphoma, and immunodeficiency states. So this process, no, or procedure is based on light scattering derived from suspension of particles of a flowing stream of fluid, no? So yung fluidics kasi is one of the components of flow cytometry, no? So yung mga cells, they pass through, no, a certain aperture, kumbaga. Okay? So very important dito yung ating mga detectors, no? It's important for us to know kung ano daw ba yung nagme-measure ng cell size, ano yung nagme-measure ng internal complexity or yung granularity ng cell. So pag cell size, that would be the forward scatter. Okay? If it measures the cell size, that is the forward scatter. Okay? If it measures cell internal complexity and granularity, that is the side light scatter. Yeah. And of course, we are using fluorescent labeled antibodies, no, in order for us to quantify cell subpopulation or cell components. Okay, fluorescent antibodies na lang yung isulat ko. But these are antibodies with label na fluorescent, na fluorophore. And sino ba yung mga fluorophore na usually ginagamit natin? We have FITC, TRITC, and phycoerythrin. Okay, so the specimen for this procedure could include blood, bone marrow, and other body fluids such as aspirates and tissue sample. And our anticoagulant na pwedeng gamitin is EDTA. Take note lang that EDTA is the anticoagulant of choice for blood samples. Okay, so EDTA is the anticoagulant of choice for blood samples. However, no, pag gumamit ka nito kasi ng EDTA for your blood samples, no, you have to process it within 30 minutes. Okay? Kailangan mo siyang i-process within 30 minutes. Okay? Whereas, no, pwede tayong gumamit ng heparin also because heparin could improve, no, the stability of the sample, okay, up to 24 hours. Okay, improve the stability of the sample, okay, up to 24 hours. Okay, then this procedure requires removal of red blood cells, no, prior to testing. The very reason here is that yung mga red blood cell kasi natin, most of the time halos kasing laki ng mga lymphocytes. Okay, so we have to remove the red blood cells prior to testing. We could use the density gradient centrifugation method, which makes use of fecal hypic solution. And if we use this, no, nagkakaroon guys ng tatlong layer. Ano yung layer na to? We have the plasma on top. Okay, we have the plasma on top. Okay, here. Then we have the mononuclear cells in the middle. We have red blood cells, granulocytes at the bottom. So syempre, kukunin natin or i-harvest natin yung ating lymphocytes from the middle because lymphocytes are mononuclears. Okay? However, the problem with this, no, with the density gradient centrifugation method is that it is time consuming, no? Okay, it is time consuming and this could also result to selective loss. Okay, selective loss of some cell population. Okay, that's why, no, the preferred method, no, of removing red blood cell is the erythrocyte lysing agent. This is which makes use of ammonium chloride. So this is the preferred method. Okay, i-lyse mo na lang yung mga red blood cell. Okay, so we now have the human leukocyte antigen or otherwise known as the major histocompatibility complex. Okay, so the gene, okay, or the set of genes, no, that is responsible for the expression of these antigens are located, no, in the short arm of chromosome 6. Okay, the short arm of chromosome 6. And these HLA antigens are inherited as haplotype from each parent in a simple Mendelian inheritance pattern. So pag ganito guys yung manner of inheritance ng HLA antigens na namamana siya bilang haplotype. So that means, no, that there is a 25% chance that two siblings will be HLA identical, no? So there is 25%, okay, 25% chance that two siblings, okay, will be HLA identical. Okay, so this is again otherwise known as MHC, okay, or major histocompatibility complex. Okay? So we have class 1, 2, and 3. Okay, unahin ko muna yung class 3 antigens natin. So yung class 3, they are not expressed on the surface of cells. So take note ha, yung class 3 MHC, they are not expressed on the surface of cells. Rather, they are being secreted, no, in the form of proteins, no, certain proteins or certain cytokines. No? So example, no, of these are your C4A, no, C4A, C4B, C2, no, and factor B, no. These are complement proteins, and example of a cytokine, no, that is coming from, no, yung part of our class 3 HLA would be the tumor necrosis factor, no, yung TNF. Okay? So yan. Now, for class 1 and class 2, so class 1, take note natin muna yung major genes niya. So we have HLA, A of course, no? So sulat ko na lang B and C, no? So we have HLA-A, HLA-B, HLA-C. And take note na itong mga HLA na ito, meron silang mga multiple alleles, no? So each locus, kasi tawag natin dito locus eh. So HLA-A, no, that is a single locus. Okay? So meron siyang mga multiple alleles within that locus. So pwede mo siyang tawagin, let's say for example, HLA, pwede mo siyang tawagin HLA, okay. HLA-A 01, pwedeng HLA-A02, no? So these are specific alleles, no, located inside, no, inside the HLA locus, no? So take note, the major locus, no, for the class 1, we have HLA-A, B, and C. So meron din tayo mga minor genes or minor locus, no? So tinawag sila guys na minor locus because yung kanilang degree of polymorphism is medyo mababa. So hindi sila ganoon hindi sila ganoon ka-variable kung baga. Ito mga minor genes, no? So that includes HLA E, HLA-E, we have HLA-F and HLA-G. Okay? Now, for the structure naman of our class 1, so take note that it is a heterotetramer, no? Kasi apat yung component ng structure. So we have 3 alpha chains, no? Meron siyang 3 alpha chains plus a beta 2 microglobulin. Sorry. Okay, we have 3 alpha chains, okay, plus beta 2 microglobulin. Okay? For the cell distribution, alam naman natin that class 1 antigens are found in all nucleated cells. Okay? And the size of the peptides that they are usually binding is composed of around 8 to 11 amino acids. The nature of the binding of the peptide binding clef is close at both ends. And it interacts, of course, with our with our T cytotoxic cells. No, so the nature of the antigen that it presents, no, is endogenous. No, so it presents, okay, endogenous antigens, okay, to CD8, no, CD8 T cells. No, so that means, no, ito yung ating mga T cytotoxic cells. Okay? Now, yung class 2 naman natin, yung mga major genes niya includes HLA-DP, okay, DQ, and DR. And katulad din no ng ating HLA ng ating class 1, meron din siyang mga alleles within a specific locus. Okay? And meron din siyang mga minor genes. The minor genes include HLA-DM and DO. So for the structure, katulad din ng ating class 1, this is also a tetramer. It is also composed of 4 units, but the units here are 2 alpha chains. Okay, 2 alpha chains plus 2 beta, no, beta chains. Now, if class 1 are found in all nucleated cells, our class 2 antigen is only found in antigen-presenting cells. No? So nakikita sila sa ating mga antigen-presenting cells, sa ating mga macrophages, okay, macrophages, monocytes, ano pa ba? B cells, no? Yung B cells. Di ba kanina? Yung developing B cells, merong MHC class 2. Diba? We have our dendritic cell, kasi this is also an antigen-presenting cell. Okay, dendritic cells. And we have our thymic epithelial cells. Okay, thymic epithelial cells. So these are the cells that are usually possessing MHC class 2 in their surface. Okay? The size of the peptides, no, na binabind ng class 2 is around 13, okay, to 18 amino acids in length. And the nature of the binding clef is open at both ends. Okay? Now, yung interaction naman ng class 2 natin with the T cells is that it presents, okay, it presents exogenous naman, no? Kanina, no, sa ating class 1 endogenous, dito exogenous, okay, antigens. Okay, it presents exogenous antigens, okay, to CD4 T cells. So yan yung ating human leukocyte antigen. Now, so what are the functions and applications, no, ng human leukocyte antigen or ng ating MHC? Remember na kino-consider sila na risk factors for certain autoimmune diseases and sometimes, no, yung mga drug hypersensitivities. So meron kasing mga disease kumbaga na namamana through these particular locus or loci, no, ng mga MHC. Okay? So we have here a list, no, of diseases and drug hypersensitivity that are associated with certain HLA loci. But other than that, yung HLA natin is importante din in mounting immune responses. Okay? Kasi it influence yung pagiging antigenic ng isang substance, 'di ba? It also is useful, no, in determining histocompatibility as well as in paternity testing. Okay? So, punta tayo, no, sa mga disease. Number 1, yung ankylosing spondylitis. It is associated with HLA-B27. Behçet's disease is associated with HLA-B51. Celiac disease is associated with HLA-DQ2 and DQ8. Narcolepsy, specific yung narcolepsy, no, DQ B1, O6, O2, no? So, kumbaga this is the specific, no? Ito na yung pinaka-specific na location talaga, no, sa chromosome 6, no, kung saan nakikita yung gene, no, that could code for narcolepsy. Then we have rheumatoid arthritis that is associated with BR, B1, O4. We have abacavir sensitivity. That's HLA-B 57, O1. And we have carbamazepine sensitivity. We have HLA-B 1502. Okay? So other than this, syempre no, include na lang natin yung si SLE, no? Kasi si SLE favorite din ito sa ASAP exam. Okay, so for SLE is associated with HLA-DR2, okay, and DR3, okay? Then we have our autoimmune thyroid conditions. Okay? Autoimmune thyroid conditions, no? So sino sila? Yung si Graves disease. Okay, Graves disease. Saka yung isa si Hashimoto's. Okay, Hashimoto's thyroiditis. Okay, so these are autoimmune thyroid conditions. Associated din siya, no? Similarly with SLE, associated din siya with HLA-DR3. Okay, then we have alopecia areata. Okay, which is associated with HLA-DR5. Alopecia areata. Okay, HLA-DR5. Okay. So far, no, that's it for our human leukocyte antigen. No? Pero meron din itong application sa blood bank, no? Kasi in blood banking, we will be talking about minsan kasi nagpo-produce tayo ng mga anti-HLA antibodies, di ba? Actually, in my exam, I have a lot of parang mga case studies wherein parang for example, no, yung anti-HLA kasi na antibodies, no, if you have anti-HLA antibodies, usually that would result, no, to platelet refractoriness, no, yung you are transfused, 'di ba, for a certain unit, no, of platelet, 'di ba, we are expecting a certain increment in our platelet count after, let's say, for example, no, kung random donor platelet 'yun, dapat we are expecting na tataas yung platelet count natin by 5,000. So parang I had a case study na parang 8 units na yata yung nabigay sa kanya. However, the increment is not it does not match, no, with the expected increment. Tas yung tanong, ano daw yung possible reason? So isa sa pinaka-common talaga na cause of platelet refractoriness is whenever you are developing already anti-HLA antibodies. Okay, anyway, we are to discuss that further, no, sa blood bank. Now, let us proceed, no, sa mga immunoglobulins or antibodies. No? So immunoglobulins or antibodies, these are glycoproteins. Ibig sabihin, they are made up of protein and carbohydrates, and they are constituting, no, for approximately 20% of our plasma proteins. Okay? So lahat ng mga immunoglobulins, again, are glycoproteins because they are made up, no, 86 to 98% of them, of their structure is made up of a polypeptide. Okay? Polypeptide. And 2 to 14% is carbohydrate. Okay? So it migrates at the gamma region when serum is subjected to electrophoresis at a pH of 8.6. That's why formerly, tinatawag din itong mga immunoglobulins as gamma globulins. And again, our antibodies, no, are the major humoral component of the adaptive immune response. Okay? So first, pag-usapan muna natin, no, yung structure ng ating antibodies. So our antibodies are made up of a basic 4-chain polypeptide structure. So ibig sabihin ano siya? Tetrapeptide siya that is composed of 2 identical heavy chains and 2 identical light chains. Itong mga heavy chain, this usually dictates the class of the antibody. So we have 5 classes of antibodies corresponding to the 5 type of heavy chains. What are they? We have the mu, the gamma, the alpha chain, the epsilon, okay, and the delta. So, ibig sabihin kung IgM yung kanyang heavy chain mu, kung IgG gamma chain yun, kung alpha that's IgA, epsilon for IgE, and delta chain, no, for IgD. Then we have 2 identical light chains. So the light chains could either be kappa, no, kappa, or pwede ding lambda. Okay? Kappa, no, or lambda. So yung kappa na usually ito yung common kasi nasa ano siya, nasa 65%, no? 65% of our antibodies yung light chains nila kappa. And nasa 35% lang yung lambda. Okay? Emphasize ko ulit ha, sa isang antibody, hindi pwede na meron siyang kappa chain and lambda chain at the same time. Okay? That's abnormal. So it's either na meron lang siyang kappa chain or meron lang siyang lambda chain because again, it has to be 2 identical light chains. Okay? Now, these heavy and light chains are held together by non-covalent forces and disulfide interchain bridges. No? So I would like to emphasize here that these disulfide bridges can only be between 2 heavy or between a heavy and a light chain. No? So dapat yung disulfide bridge dapat between 2 heavy chains lang or between a heavy and a light chain lang. Dapat walang disulfide bridge between 2 light chains because that is also abnormal and that is found no sa Benz-Jones protein. Okay? So heavy tas heavy light lang dapat yung connection. Okay? Now, each chain has a variable region and 1 or more constant region. Now, for the variable region, that is actually no, ito yung variable region eh. Ito siya. Yan, that's the variable region. So, for the variable region, that is basically no the first 110, no? The first 110 amino acids. Okay, the first 110 amino acids of both chains. Okay? So yan yung nagdidikta sa variable region. So from the name itself, variable, ibig sabihin iba-iba, no? Magkakaiba, no, yung mga components ng amino acids dito. However, in the constant region, so basically if they are belonging under the same immunoglobulin class, the constant region would be the same also. Okay? So hindi pwede na pareho kayong IgG pero yung laman ng constant region nyo hindi pareho. Okay? Although meron ding antibody diversities, no, that we are to discuss in a while. Pero minor lang yun eh, minor lang yun ng mga differences, no? So majority pa rin talaga no na component ng constant region should be identical within an immunoglobulin class. Okay? So also meron tayong tinatawag na hinge region. So take note lang of the location of the hinge region that is between C1, yes, tama no, that is between C1 and C2. Okay, that is between C1 and C2. And remember that the hinge region is proline rich. Okay? This is proline rich. Kasi yung proline, this is responsible for the antibody flexibility. Okay? Then of course, no, yung ating antibody, meron din silang mga receptors, no? The receptor that can be found in the CH2, okay? In the constant region 2. This is where we could find the receptors for complement proteins. Okay, and in the constant region 3, okay, this is where we could find receptors for phagocytes. Obviously, no, yung ating CH2 tsaka CH3, they are now part of the FC region. Okay? So that is the complement and the FC receptor binding region of our immunoglobulin. Okay? So take note, emphasize ko ulit, yung ating antibody is a tetrapeptide structure. It is composed of 2 heavy and 2 light chains. Yung light chains natin, meron siyang dalawang domain. Okay? Yung light chains, meron siyang dalawang domain. No? Ito yung light chain. Okay? Dalawa yung kanyang domain. The variable light and the constant light. Okay? Now, yung ating heavy chain, no? This one yung nasa loob, ito yung heavy chain kasi. Meron siyang 4 to 5 domains, no? 4 to 5 domains. Okay? Isang variable heavy, tapos pwede kasi guys na 3 or 4 yung constant heavy domain. Okay? Pwede kang magkaroon ng CH4 pa dito. Okay? So, that's it, no, for our immunoglobulin structure. So again, emphasize ko ulit yung ating disulfide linkage, it should be interchain or intrachain lang. No? So pag sinabi nating intrachain, that is between 2 heavy chains only. Pag sinabi nating interchain, that is between a heavy and a light chain. Okay? Hindi ka pwedeng magkaroon ng disulfide bond between 2 light chains because that is abnormal and is only found sa abnormal din na protein, which is Benz-Jones protein. Okay? Now, let us talk about the 5 immunoglobulin classes. Okay, so these are the forms that are found in the blood. We have IgG, IgA, IgD, and IgE. These are monomers. IgA that is found in the secretion, yung dimer, and IgM is a pentamer. So kung monomer siya, ano yung kanyang valence? Okay? So yung valence usually it corresponds with the antigen binding site. So kumbaga yung valence it represents yung kamay ng immunoglobulin mo. So if you are a monomer, dalawa yung kamay mo. So your valence is 2. If you are a pentamer, okay, your valence is 10. And yung valence kasi guys ng immunoglobulin, no, later on when we are to discuss about serologic reactions or agglutination precipitation reactions, the valence is also important when we talk about avidity of our antigen-antibody reactions. Okay, so syempre pag mataas yung valence, no, the avidity will also be higher. Okay? Then for the average concentration, okay, syempre pinakamarami sa dugo natin is yung IgG. Okay? Followed by IgA, then we have IgM. Okay? Actually yung mnemonic dyan is GAMDE, no, GAMDE. Okay? But I took this table, no, from the ASCP review book. So hindi naman nila in-emphasize na yung levels ng IgD tsaka IgE, but if I am to take it from Stevens, parang nasa 0.2% la, 0.2 milligrams per dl lang yung IgD, tapos parang 0.02 yung IgE. No? So sobrang konti sila. Okay? Next, for complement fixation, IgG and IgM, yes, they could fix the complement specifically, no, for the classical cascade. Yung IgA, yes, it could also fix the complement, pero alternative lang. Take note of this alternative cascade lang yung kayang i-fix ng ating IgA. Okay? Then, notable properties, no, ng ating mga immunoglobulin, yung IgG, of course, it is the only immunoglobulin that could cross the placenta. Yung IgM natin is the first immunoglobulin made by a developing fetus and naive B cells. Remember, di ba, na yung IgM natin is the first surface immunoglobulin. Okay? It binds B cells and it is also hemolytic, yung ating IgM. Yung IgA natin is the most abundant in the saliva, the tears, and other secretions. Kasi basically, siya lang naman yung nakikita most of the time sa secretions in a dimer form. Yung IgD natin has a role in B cell development, and yung IgE natin has a role in allergies and parasitic infection. Yung IgE kasi is often found in the surface of our in the surface of our basophils and mast cells. Diba? Nandiyan guys, so it is bound to mast cells and even our basophils. And whenever an allergen is attached to it, no, nagkakaroon ng degranulation ng ating mga mast cells and basophils, releasing no yung mga active or yung mga active substances that is responsible for hypersensitivity reaction. Okay? So, yan. Now, an immunoglobulin, no, yung isang antibody, pwede din siyang maging antigen. Okay? So we have an immunoglobulin classification based on the location of antigenic determinants or determinates no, of our antibody. Again, this is the classification of antibodies if we are to consider them as antigens. So first, different classes based on the type of heavy chain, that is the isotype. Okay? The isotype of our immunoglobulin. So, yung basically the class, no, the class of immunoglobulin, that is the isotype, IgG, IgM, and so on. Okay? Next, minor variations present in some individuals and not present in others. This is formed by constant regions of heavy and light chain. That would be allotype. Okay, allotype. Epitope formed by the variable regions of heavy and light chains. That would be idiotype. And lastly, antibody formed against the idiotype. So obviously that is an anti-idiotype. Okay, anti-idiotype. Okay, so ituturo ko kung alin dito yung isotype, alin dito yung allotype, alin dito yung idiotype. Balik tayo dun sa antibody structure natin. Okay? So first, no, yung isotype. Okay? So yung isotype is nakabase siya sa ating heavy chain. So basically, itong blue na area, okay, that dictates the isotype of your immunoglobulin. Okay? That's the isotype. Next, ano yung kasunod doon? Allotype. Yung allotype is based daw on the constant region, both of the heavy and the light chain. So, ibig sabihin, pag allotype yung pinag-uusapan, ito yung kanyang determining factor, itong area na to. Okay? Yung constant region ng parehong light tsaka heavy chain. That's for the allotype. Yung idiotype naman is the variable region daw of both the heavy and the light chains. So pag idiotype, ito yung nagiging basis, itong area na to. Okay? So yan siya. No? So nagkakaroon ng pagkakaiba, nagkakaroon ng mga minor differences in those areas. Kaya pwede ding mag-represent na parang iba't ibang uri ng antigen itong ating mga antibodies. Again, ulitin ko this is for the isotype. Okay? This is for the allotype. Diyan yung differences. And lastly, this is for the idiotype. And kung meron kang antibody that is attacking the idiotype region, then that is an anti-idiotype. Okay? Now, let's talk about antibody response. Okay, so we have the primary and the secondary response. So the primary response obviously is happening when during the first encounter of our immune system, kumbaga, no, to a specific antigen. And this is occurring primarily, no, this one is occurring primarily in our lymph nodes and spleen. Okay, lymph nodes and spleen. Okay? Now, for the primary response, yung punto dito, guys, first time mong ma-experience, first time mo siyang ma-encounter. So, kumbaga kinikilala mo pa lang yung antigen. Therefore, the lag phase, no, the lag phase here is longer as compared to that of the secondary response. Usually, no, yung antibody production mo, no, nagpipeak pa siya, no? Usually yung antibody production mo nagpipeak siya. Sorry, lymph nodes. Okay? Usually, no, the antibody production peak after 7 to 10 days. Okay? So matagal, no? Matagal yung lag phase nito. And of course, the antibody titer is lower. Okay? Lower yung antibody titer. And the nature of the antibody that is produced, no, predominantly is IgM. No? So you have large amount of IgM. Okay? Large amount of IgM. Okay, large amount of IgM and a small amounts, okay, of IgG. Yan yung napoproduce. So that makes, no, yung ating IgM as the predominant, okay, the predominant antibody in the primary immune response. Okay? So take note, no, that in the primary immune response, it involves both our B and T cells. Okay? So pakidagdag na lang dyan, it involves, okay, both B and T cells. Okay, now, for the secondary immune response naman, ito yung tinatawag natin na anamnestic response. That means it is the second time around na encounter kumbaga natin, no, yung isang antigen na expose tayo before. Okay? And siyempre, no, we are to expect that the response would be faster as compared to that of the primary kasi kilala mo na yung kalaban eh, 'di ba? Now, this secondary response is occurring primarily naman in the bone marrow. Okay? It occurs primarily in the bone marrow and later, no, later in lymph nodes and spleen. Okay, so it occurs primarily in the bone marrow and later in lymph nodes and spleen. Now, again, mas mabilis ka nang mag-respond this time kasi kilala mo na. Therefore, the lag time is shorter and you are able to reach, no, the peak of antibody production within 3 to 5 days. No? Nakakapag-peak ka na ng antibody production within 3 to 5 days. And the titer, no, or the amount of the antibody that you are able to produce is higher na, no? So increase na yung titer mo, and the nature of the antibody that is produced is predominantly IgG, no? So you are able to produce large amounts of IgG, okay? Small IgM, okay, small IgM, IgA, and IgE. Okay? So large amounts of IgG, small IgM, IgA, and IgE. So take note, IgG naman yung predominant antibody natin in the secondary response. And take note, in the secondary response, this one is no longer dependent on T cells. So that means it does not involve T cells anymore. No? So not dependent on T cells. Okay? Now, of course, meron tayong tinatawag, no, na antibody fragmentation. No? I failed to include that, no, in the notes. Pero segue lang natin, dagdag natin yung antibody fragmentation. The method depends on the type of antibody, of course. If you are to digest, kumbaga, no, yung IgM, okay, let's say, sorry, antibody fragmentation. Okay? So if you are to digest, for example, yung IgM, of course, your target is to break the J chain. Okay? You have to break the J chain because that is the one that connects, no, the monomers of your IgM. So ano yung mga pwede natin gamitin dito? We could make use of DTT. okay, or dithiothiotol, okay, or pwede din tayong gumamit ng 2-mercaptoethanol. Okay, 2-mercaptoethanol. Yan. Okay? Now, if we are to break naman no, yung ating IgG, or let's say yun, no, yung mga monomers, okay, yung mga monomers, if we are to fragment, if we are to break them no, into several fragments, meron tayong dalawang techniques. No? We have the use of pepsin, and the other one is the use of papain. So, pag gumamit tayo ng pepsin, okay, take note that pepsin, okay, cleaves, okay, the antibody below. Take note it cleaves the antibody below the hinge region. And because of that, no, it will only result to 2 fragments. No? Dalawa lang yung fragments nito. Okay, ng pepsin digestion. Okay, the 2 fragments are, you will have one FAB2 fragment, no? So you will have one FAB2 fragment and you have one FC fragment. Okay? Now, if you use papain as your digestive agent, no, for this, papain cleaves the antibody above the hinge region. Okay, it leaves the antibody above the hinge region. So as a result, you will have 3 fragments. Okay, the fragments are, you'll have 2 FAB. Okay, you will have 2 FABs and you have 1 FC. Okay, so yan naman yung papain digestion. Actually, ituro ko na lang din to kasi ito yung ginagamit na palatandaan. Pag pepsin daw, dalawa lang, pepsin, kasi dalawang syllables. Pag papain daw, basahin mo siyang papain, no? Papain, para tatlo, no? Tatlong fragments yung napoproduce. Okay? So yan yung fragmentation no, ng antibodies. Okay? So now let us proceed no, sa complement. Okay, so the complement is also a component of our of our humoral immunity that is both for the innate and the acquired. Kasi we have the alternative cascade as part of our natural immunity, and we have the classical cascade as part of our acquired immunity. So the complement is a series of more than 30 soluble and cell-bound proteins that interact in a very specific way, okay, to mediate and enhance host defense mechanisms against foreign cells. So ibig sabihin guys, cascade nga siya, di ba? So the activation of one complement component, for example, or complement protein results in the activation of the other. Okay? Now, yung complement proteins natin are heat labile substances that are present in normal non-immune serum. So heat labile sila, nasisira sila ng heat. That's why meron tayong tinatawag, 'di ba, na procedure na heat inactivation, no? Or yung tinatawag natin na complement inactivation, wherein we are heating the serum sample at 56 degrees Celsius for 30 minutes, 'di ba? Para ma-inactivate yung complement. And most plasma complement proteins are synthesized in the liver also. Okay? Except for the C1 components that are synthesized no in our intestines, no? Synthesized by the intestinal epithelial cells, and yung factor D, no, that is produced by the adipose tissue. Yeah. So but the rest are produced in the liver. So most of these proteins are inactive precursors. Ibig sabihin mga zymogen sila. And whenever they are converted to their active enzymes, they will now activate the next, no, sa kanilang cascade. Okay? So, very precise, no? Very precise dapat yung order nito ng complement and activation. And usually, no, the order is divided into 3 phases. What are they? First, yung tinatawag natin na recognition. Okay? So, first is the recognition. So, let's use the classical cascade as an example. No? So for the recognition, usually this involves the attachment, no, of the complement protein sa antibody na nakadikit sa target na cell. So usually, no, this is made possible by the C1 component, no, of the classical cascade. To be specific, si C1q talaga yung tinatawag na recognition unit. Okay? So si C1q, siya yung dumidikit doon. If this is the target cell, di ba? You have your, let's say, you have here your attached immunoglobulin G. No? Si C1q, siya yung dumidikit dito sa FC portion. Okay? So may didikit din sa isa, for example. Okay? So siya talaga yung taga-recognize. Okay? So that is the first step, no? Recognition. Then that would be followed by activation. Okay? So in the process of activation, this often involves, no, the cleaving, okay? The cleaving of C4, C2, and C3 into its A and B components. 'Di ba? Kasi pag na-cleave natin yung C4, magkakaroon tayo ng C4A, C4B, 'di ba? So that is in the activation stage, no? The cleaving of C4, C2, C3 components into A and B, okay, components. Now, remember na itong mga A and B components na ito, no? Some of them will function as anaphylatoxins, some of them will function as opsonins, okay? And some of them will take part, no, in forming a complex, a certain enzyme, no, that will also activate the next, 'di ba? That's why meron tayong mga tinatawag na mga convertase, no, sa complement cascade. Okay? So eventually, no, because of the cleaving of C4, C2, C3, eventually makikleave na yung C5. And the cleaving of the C5 is the start, no, of the membrane attack complex formation. So that is the final stage, no, wherein it is initiated, no, initiated by C5. Okay? And when C5 is cleaved, no, nagiging C5A siya, tsaka nagiging C5B. Yung C5B, no, didikitan ni 6, 7, 8, and 9. This is now your membrane attack complex. And the very purpose of this is cellular lysis. No? Nagkakaroon ng cellular lysis. No? So that should be a very precise manner, no, yung activation niya. Okay? So yung manner of activation also nagkakaiba-iba yan depending on the cascade. No? And we have 3. Okay? We have the classical, the mannose binding lectin, and the alternative cascade. Now, pinagtabi ko guys yung classical at mannose binding lectin kasi halos pareho yan sila. Nagkakaroon lang ng difference in the recognition unit. Okay? So, in order for the classical cascade to be activated, ano ba yung kailangan? Dapat meron tayong antigen-antibody complexes. Okay? Dapat merong antigen-antibody complexes. Now, for this, di ba, in order for the classical cascade to be activated, kailangan mo ng dalawang IgG na naka-attach dun sa antigen. Pero pag IgM lang, you only need one, no? In order for the classical cascade to be activated. That's why nga, di ba, yung IgM, siya yung kino-consider as pinakamagaling when it comes to complement activation. Okay? Now, the order of activation dito, no, for the classical cascade would be C1. Okay, C1. Then we have C4, C2. Okay, para mabuo yung C3 convertase, para i-cleave yung C3. mabuo yung C5 convertase, i-cleave yung C5, tapos didikitan na siya ng C6, okay, C7, sulat ko na lang 6, 7, 8, 9, no, para mas madali. C2, C3, then 6, 7, 8, 9. Kasi yung 6, 7, 8, 9, hindi mo naman na sila kinikleave eh. Di ba? Now, the C3 convertase for this is C4b, 2A, and our C5 convertase is C4b, 2A, 3B. Yeah. Basically, that is similar with the mannose binding lectin. It's just that the requirement for activation here is the presence of mannose. Mannose is a sugar that is usually present no, sa mga cell wall ng mga bacteria. Okay? Then, instead of having the C1 component, yung ating mannose binding lectin, meron silang mga MASP. No? Yung MASP, ito yung counterpart ng C1, no, ng classical cascade. Then the MASP, okay, will activate C4 and C2 simultaneously, eventually activating C3, activating C5, no? Tapos didikit na si 6, 7, 8, tsaka si 9. So halos pareho lang talaga, no, yung ating mannose binding lectin tsaka yung ating classical cascade. They only have their difference in the recognition unit. That's why pareho din sila ng C3 convertase. Yung C3 convertase ni MBL is also C4b2A and yung kanyang C5 convertase is also C4b2A 3B. Okay? So yung medyo iba lang talaga, okay, is the alternative cascade. And ano ba yung mga kailangan, no, in order for the alternative cascade to be activated? Yung pinaka-stimulus talaga, no, the major stimulus, no, for the activation of the alternative cascade is yung mga bacterial lipopolysaccharide. Okay? Bacterial LPS na lang, no, lipopolysaccharide, and even no or other bacterial products. Okay, or other bacterial products. So whenever they are present, no, nagkakaroon ng alternative cascade activation. So if you would notice, it does not require antigen antibody. That's why it's part of the natural immunity. Okay? So, other than that, no, ano pa yung possible, no, na activator ng alternative cascade? Guys, na-mention natin to kanina, yung IgA, no? The IgA binding, no? Because yung IgA can also fix the complement pero alternative lang. Okay? Now, isa sa pinaka-characteristic ng alternative cascade is that parang binabypass niya yung recognition unit, no? So wala siyang C1, wala siyang C2, saka C4 involvement, no? So binabypass niya, diretso siya sa C3, okay, activating C5, okay, and eventually, no, 6, 7, 8, 9, no? Binabypass niya, no, yung ating recognition unit, which is the C1, which is composed of the C1 components, no? However, no, kung meron tayong tatandaan dito yung pivotal point ng lahat ng cascade na to. Lahat ng cascade na to still, okay, involves C3. Okay? That's why, no, if we are to discuss about our if we are to discuss, no, yung ating mga diseases, no, of the immune system, okay, ay yung pinaka-delikado or pinaka-severe na complement deficiency would that be of C3. Kasi pag wala kang C3, hindi ka talaga makakamount ng complement activation kasi it really requires C3. Okay? Now, for the C3 convertase of alternative, that is C3b, BB, and yung kanyang C5 is C3b, BB, 3B. Yan. Okay? Now, tatandaan lang natin dito no, that there are other products also of the complement activation that functions as opsonins, anaphylotoxins, and chemotaxins. No? Sino-sino sila? Sino ba yung nagfa-function ng opsonins? Okay? So yung opsonin natin would be C4B. Okay? C3B rather, no? C3B. Okay? Then we also have our chemotaxin or chemoattractant. Ibig sabihin, no, it attracts phagocytes no towards the site of inflammation. That would be the C5A no component. Then we have anaphylotoxins. So pag sinabi nating anaphylotoxins, they are able to result to degranulation of mast cells and basophils. No? So parang allergic reaction, anaphylaxis nga, di ba? So sino-sino sila? We have C5A, C4A, and C3A. Okay, so dito makikita natin that C5A pala functions both as an anaphylotoxin and a chemotaxin. Yan. Okay?